Good evening. Thank you for coming. I'm Stewart Brand from the Long Now Foundation. I should say that most of you have these question cards, and in the semi-darkness of the theater, it's helpful when you've written out a question to come up to the front for Kevin Kelly and me to look at to later address the speaker with. It's helpful if you wave it so that the guys in the yellow hats can see it, come collect it, and bring it up. And write questions anytime during the talk, during the Q&A. That's the interactive part. Now, the Long Now Foundation is called the Long Now because we have in mind the next 10,000 years and the last 10,000 years. And Peter Schwartz here is the one who came up with that number because 10,000 years ago, there was a biotech revolution having to do with mainly plants and then animals that we eat and use. We started genetic modification, genetic engineering in a big way, and humanity took a swerve that it's still getting used to. And then about 150 years, another biotech revolution happened with biomedicine, and humans started to be able to control their health, be able to control their birth rate, and then we get to the 21st century, which is regarded by many as the century of biology, which to a large extent means the century of biotechnology. And so these revolutions that we've seen seem to be on a kind of a biotech Moore's law of getting more and more frequent and happening now in one lifetime instead of it a millennia at a time. And the current acceleration of genetic science and genetic engineering is, as often the case, being carried by very sharp individuals, scientists who find the right problems, find the right people to work with, find the right funding, and swarm ahead. And really the exemplary case of that these days is Craig Venter. Please welcome him.
Well, thank you, Stewart. It's certainly nice to be here with the Long Now Foundation, taking a long view, hopefully forward with humankind. I think it's very much an open question, but we're trying to see if we can change some of the equations, maybe to help that process along. I'm going to talk about two different phases of information tonight. The first phase is gathering the information. I actually refer to it in my case as reading the genetic code. And then I'll turn to how now we're using that information to change evolution, to change biology, to change hopefully some parts of society with writing the genetic code. And I'll talk about the early steps of that. All this has happened in a pretty short period of time. We sequenced the first genome of a living species, Haemophilus influenzae, in 1995. Before then, we developed techniques for scaling up gene discovery, but we've only had complete genomes of living organisms for a relatively short period of time. It's changing our view of looking at biology. We did two genomes in 1995, and I'll talk about the second one in the later phases of this. Had very different size genomes, very different characteristics. But after those first two that we had to fund ourselves, we started getting a lot of government funding to scale up the process. We got funding to do almost every major human pathogen. We moved into plants, then simple animals, then more complex ones such as fruit flies and people. The big change in technology after 1995 came surprisingly only 4 to 5 years later with the fruit fly genome. Haemophilus was 1.8 million letters of genetic code, Drosophila 180 million, and they were both done in the same 4-month time period. And 9 months later, we had the 3 billion letter first draft of the human genome. Computational aspects were very important. We actually had to build the third largest computer in the world to do this calculation. Now it's not so hard to do with much more standard computers. 1.5 teraops in 1999 was a huge computer. Today, we have things 10, 20, 30, and 100 times that size. Just this last year, we published the first complete human genome. This was the diploid genome, so it had both sets of chromosomes from both parents. What was done in 2000 and 2001 ended up being only half the job. The public effort only got it half right because they set out to only do half, thinking the other half would be easily discernible. The project at Celera actually tried to do the complete genome, but we made the mistake of overreaching. We tried to do the genome of 5 individuals. It was 3 men and 2 women. No, it was 3 women and 2 men. Sounds like a San Francisco movie. But because of the diversity, when we assembled the genome, we ended up subtracting out all the major variation between them. So the message, and many of you will remember it, 2000, 2001, is we all have the same set of genes, and we all differ from each other only in 1 out of 1,000 letters of genetic code. And this started all kinds of people down certain tracks. We have a pretty large industry now just to measure those 1 out of 1,000 differences. But it turns out we're all 1 to 3% different from each other. So it's a much greater variation than we imagined. To put that in context, we thought we differed from chimpanzees only by 1.27%. So you're probably hoping that number changed as well. Or we really have some explaining to do about evolution. It turns out we're 4 to 5% different from chimps. And so this individual variation was totally missed. The 2 sets of chromosomes that each of you have, each of those sets of chromosomes, they don't necessarily have the same gene that the other pair has. For example, there's a major gene associated with detoxifying environmental toxins that of the Caucasian population, a third have no copies of this gene, another third have 1 copy, and another third have 2 copies. But even when you have 2 copies, there's major variation in these. So it's hard to find 2 of us with the exact same genetic code, and we have a lot of complexity. I think it's important that we know it now because it means we can move towards getting the right answers. In this first genome, almost half the genes had a major variant in them, and this certainly is going to confound anybody who was hoping to get simple interpretations out of the human genetic code. So we have all kinds of companies promising that now for $1,000, but it's only a partial peek at what might be there. 75% of the letters of genetic code that are variant between any 2 of us didn't show up in the single nucleotide polymorphisms that people were measuring. So we're scaling up now to do 10,000 human genomes over the next decade. Hopefully it won't take 10,000 years, Stewart, but maybe only 10. And we will finally have the first chance to understand what's genetic, what's nature, and what's nurture. Not only with the genetic code, but we're trying to collect extensive phenotypic information on all these individuals that we're going to be sequencing. And that's a much greater challenge when you think about all the information that might describe you or your lives, how you look, how you think, your body structure, your organ structure, your metabolism. All those things differ dramatically with each of us. And trying to describe that makes sequencing the genome look like the easy part. We published our data in the public access of Journal of Biology. You can download this 300 megabyte file if you want to. It's a zoomable version of the human genome that shows the types and extent of human variation. And hopefully in a short time, you'll be able to get the map of your genome this way. Working with the X Prize Foundation, we now have a $10 million prize for whoever gets the technology going to the extent that we can sequence an individual genome for $1,000 in a very short period of time. So we're hopeful that we'll be giving out that $10 million in the next 5 years. If you're looking for a little spare change, you might try inventing something worthwhile. We're releasing a new browser for the genome that allows it to be looked at down to the sequence file level as people, for example, get data from 23andMe or other places. They can look at actually a complete genome to try and put it in some kind of context and find out how much is missing from those early tests. Now, when we finished sequencing the human genome, many people were hoping I would retire, but instead we looked around for what we thought were the most important projects in science to do, particularly with what the technology that we developed for reading the genetic code could try and tackle. And it was clear to us that trying to look at the environment, not only the extensive environment around us that helps influence our genes, but the broader environment, to see if we could use DNA sequence as the new tool to see who we were sharing the planet with. There's been this statistic for a long time that each milliliter of seawater has 1 million bacteria and 10 million viruses, but nobody really knew what that meant and whether there was any diversity in that. And so we decided to do an experiment in the Sargasso Sea, just taking a barrel of seawater, filtering out all the organisms in it, isolating the DNA from them en masse and sequencing it. And just from one barrel of water, we stopped sequencing after we had close to 1.4 million new genes, maybe as 40,000 new species that had never been seen before. So the technique obviously worked because, in fact, people thought we'd find little or no life in the Sargasso Sea because it was supposed to be a desert with no nutrients in it. And I'll show you why and how these organisms survived in a minute. But we decided to look further, and we started the Sorcerer II expedition. I'd always been looking for an excuse to sail around the world on my own vessel. And so we decided we would follow great scientific expeditions such as the Challenger expedition and sail around the world taking samples every 200 miles and sequencing everything we could find to see if we could generate a different view of life. And we were absolutely stunned with what we found, and that was a special issue of PLOS that was published last year. This is the route that we followed. As with the Challenger expedition, we started in Halifax, went down the eastern seaboard into the Caribbean Sea, then down between Mexico and Cuba to the Panama Canal, through the Panama Canal and down to first Cocos Island and then to the Galapagos. And that's the dots you see here in blue. That's what was covered in the special issue of PLOS of biology. And that covered over 6 million new genes, more than doubling the number of genes known from all science up to that point. And we've now been analyzing the samples from the rest of the globe. After Galapagos, we left and sailed to the Marquesas, where we were promptly arrested because there was a debate between the French government and the French Polynesia government as who had rights over the microbes that are there. And you may not understand this, but this is a physical map. If you look at a political map, in the Caribbean Sea, for example, there's no international waters. Every drop of water is claimed by one or more countries, and you need permission from them to take a scientific sample. It's okay to fish in most of those places. You can shoot marine mammals, you can do whatever you want. But if you're asking scientific questions, it's considered extremely dangerous, and you can get arrested for it. In fact, when the organisms in the ocean are out in the middle of the Pacific Ocean, they're in international waters, and they belong to nobody or to everybody. As soon as the 1 knot current that goes across the Pacific carries them into French Polynesia, all those microbes instantly become French genetic heritage. And they're willing to defend it to the extreme. We finally got out of there and took samples and spent some time in Australia, then went across the Indian Ocean. We got halfway across and we stopped in Chagos Island, which is near where our country has a B-52 air base, and we were arrested again by the British this time because they were worried we would take samples and understand science in that water. So science is something, and organisms that are greatly protected around the world, and to be able to publish this data in the public databases, we had to put geographic GPS coordinates on every DNA sequence. So for example, if somebody makes a discovery using the sequences that we found in Australia, and you want to commercialize those, you have to contact the Australian government and negotiate something. It's not clear what. So science it's much more complicated today, but the experiments were very simple. We just simply filtered seawater through different size filters. We could then just take those filters, put them in the freezer. When we got into a port, we put them in a FedEx package with some dry ice and send them back to the lab in Rockville, Maryland, where all the DNA was isolated at once from them and sequenced, and then reconstructed in the computer. And when we reconstructed it, we were amazed with what we found. For example, every 200 miles, 85% of the sequences in the organisms are unique. The ocean's not a giant homogeneous mixture. Samples that we've taken off of San Francisco Bay will be very different than the ones off of Los Angeles and off of Seattle and Oregon. In fact, if you look at the red or the dark here, if you're colorblind, is warm water. The blue or light colors is cold water. And even the 3% of sequences that assemble across more than one site, they make an absolute distinction between warm and cold water. And some of these change on an annual basis with weather. Other things that affect it are sunlight, and nutrients, but we can tell simply from a sample of seawater, looking at the DNA in that water, where that water came from in the world. And for example, with all the ships that come and go from San Francisco Bay, if a tanker comes in, offloads its cargo, it fills up its hold with seawater and will go to its next port, dump all that seawater, and take on new cargo. So look at all the ships that come and go from San Francisco Bay each day, and imagine with 1 million bacteria and 10 million viruses per milliliter, how these environments are constantly being challenged and shifted. One of the biggest discoveries we made early on was that these organisms we discovered in the ocean had photoreceptors. Molecules very similar to our own visual pigments, and almost every one in the top parts of the ocean have these photoreceptors. The blue segment at the bottom of the slide is our prior knowledge of photoreceptors before this expedition. And these are very deep branching, a lot of diversity, so instead of being a rare gene family just affecting our visual acuity, it affects much of life on the planet. We can line up all these proteins. This was early on when we only had a couple thousand of them. And why do this? It turns out there's a single amino acid residue that determines the wavelength of light that these receptors see. So then that allows us to ask some unique questions. Do we see any association with different geographic regions with the wavelength of light? And we're quite surprised, in fact, to see something that makes sense, but nobody predicted it ahead of time. For example, the Sargasso Sea, it's a deep indigo blue. The organisms that are there, the photoreceptors, see blue light. You get into coastal waters where there's a lot of chlorophyll, they see primarily green light. You get into fresh water like the Panama Canal, they see entirely green light. So there's only one letter of the genetic code that is needed to change the wavelength of light by changing this amino acid residue. And even though there's a large abundance of organisms traversing these waters, the ones that survive and grow are the ones that can get their energy directly from the sun. And last year there was a study from a Swedish group showing, in fact, these organisms grow directly on sunlight. This is not photosynthesis. This is the same biological mechanisms that you're seeing the slide with right now. You're having light hit these receptors and they transport ions across the membrane. Well, in these microorganisms, that's how they generate their energy in a low nutrient environment. So in fact, instead of low nutrients indicating a sparsity of life, this was some of the highest density of life anywhere that we'd seen because they have these mechanisms of getting energy straight from sunlight. The other thing that we found is where people thought there was a single organism, we found evidence for thousands, all related in the sense they have similar sets of genes, similar gene order, but hardly a single one. Each one of these little bars you see on here represents 900 base pairs of genetic code. One of the things we can do is take a slice out of this data anywhere and create trees to look at the relationships between these very closely related organisms. They're color-coded by site, so we can look at things like Atlantic Ocean versus the Pacific Ocean. But the bottom, to me, is the most exciting. When you look at recent evolution, there's been a switch between blue and green light for these photoreceptors 4 different times. So you can see at such selective pressure, you get a mutation and you switch from blue to green and you're in coastal waters, obviously you're going to thrive. If you switch from blue to green out in the middle of the ocean, you probably won't. You can ask basically any question of this data in a similar fashion. There's in fact a lot of questions. How much novelty is there with all these new discoveries that have been doubling the number of all genes known to science? Are they like the photoreceptors, just new members of known gene families? Or are we really making new discoveries? And if we're making new discoveries, what's the pace of these discoveries? When we looked at the data set initially, we were quite shocked to find that our database just between Halifax and the Galapagos was twice the size of all the public databases in terms of gene content. And so we did a calculation by comparing all this data to each other. It was about 1 million CPU hours. And we found out thus far in the data set there was maybe 50,000 major gene families. Well, we only have 22 to 23,000 genes, maybe 14 or 15,000 gene families. So even all of human biology represents only a subset of this data. But if we look at this as a curve, instead of the long now, we have a long tail that basically goes out approaching infinity right now with new discoveries and new gene families, with the exception of the animal world. So just to remind people, we're just in one part of evolution, even though the tree model doesn't really hold, it's useful for these kinds of diagrams. When we look at our reading the genetic code in mammals, it's basically been saturated. So sequencing another mammalian genome, your favorite pet, the species down the street, whatever, you're not going to discover any new genes. You might discover some unique combinations or spelling of those genes, but we pretty much saturated the mammalian part of the tree. But if we look at bacteria and archaea, we're in a linear phase of discovery. We don't really know where we are, we just know it's linear. our thinking is that we're in the earliest phases of this, of just a few percent at most, which means anybody can just go out to the bay or go across to the Pacific, take a sample of seawater, and make tens of thousands of species discoveries, millions of gene discoveries, et cetera. We first actually applied these techniques to the human, looking at the bacteria that are associated with us. So the microbiome is your collection of microbes. Not all of you have all these cavities, but this is no time for cavity jealousy or anything, but we can isolate organisms from these different cavities. And for example, look at the person next to you, and maybe you see them as having thousands of species in their mouth right now, or maybe you can taste your own species. On the order of 4 million genes of foreign species in your mouth, in your intestinal tract. If you have those other parts, you have those as well. So we have more bacterial cells associated with us than we have human cells. And it turns out they affect our physiology in some pretty interesting ways. There's a company in North Carolina called Metabolism that's using high-throughput mass spectrometry to look at all the chemicals in the bloodstream. And they've worked out that we as a species can maybe make around 2,400 chemicals. If you look in the bloodstream of anybody, for example, after they've had a meal, easily find about 60% of what's there is made by our own bodies. About 30% are just chemicals that came out of what we ate. So this notion you are what you eat is partly right. But 10% are bacterial metabolites. So in fact, partly we are what we feed our bacteria and what they give us. But we're dealing at any one time with hundreds to thousands of foreign chemicals circulating in our bloodstream, not to mention what we add directly by taking pharmaceutics or other things. And we have no idea the impact of these on physiology, because in fact we didn't even know they were there before. So we're looking at the complexity of the human genome and all the variations there, and what's in our bloodstream. And if we did this test on everybody in this room, we'd get a different answer for everybody based on what you ate, and also the uniqueness of the bacteria in your own guts and your mouth. This affects our chemical milieu. This is part of the environment that affects us. We're also sequencing the air genome. So, for example, in a room this size, sitting here for an hour, you would absorb maybe 10,000 different bacteria and maybe 10 times that in viruses in an hour. If you go outside, it's twice that much, so you're actually safer in here right now, depending on who you're sitting next to again, and what they're exhaling and inhaling. So we live in a bacterial milieu. If you go into the water, you're surrounded by bacteria. So if you're out swimming and you swallow a mouthful of seawater, you've just swallowed millions and millions of bacteria and viruses. The air we breathe, the soil, our own skin, our own cavities. So we are as much dependent on bacterial metabolism as human metabolism. We've been collecting all these genes, all this information, trying to understand the complexity. And we decided one way to try and understand the complexity was to try and mimic it. This is standard in chemistry to prove that you have the structure that you think you have, you remake that compound. We decided to try and do this with the genetic code, starting with some pretty simple organisms. The second organism that was sequenced in 1995 was Mycoplasma genitalium. This is its genome, at least its depiction of it. Very much unlike our genome, so it has a little over 500 genes in contrast to our 22,000. we have gaps in our genetic code with no genes that are much larger than this entire genetic code. So bacterial genomes, you can see these bars represent genes, have very little intergenic space. This is still the smallest genome of a self-replicating organism. So it'll grow on its own in the lab. They're smaller genomes, but they're dependent on having a symbiote organism for its growth. So it's not clear whether they're extension of the viral world or just truly simple organisms that are dependent on others. And we just ask simple questions. If one cell needs 1,800 genes to live, and this one needs 500, is this actually the minimal set? Can we define life in molecular terms based on the genetic code? And so we set out to try and knock out genes in this genome to see if we could get down to a smaller number. So every place you see one of these small triangles is where a transposon, which is just a piece of DNA, very much like a small virus, that can randomly insert anywhere in the genetic code. Then we select for living cells. So only genes that are not essential can tolerate transposons going into them. So this is very much a negative map. You can see some genes can take large numbers of transposons. But keep in mind, these experiments only done one gene at a time. And we got to a number of around 100 genes that we could knock out. But we doubted whether if we knocked all 100 out that we could get to a living cell. In fact, if we look at a metabolic map of this simplest of organisms, it may look complex, but this is remarkably simple from looking at any one of cells in the human body. And here's if all the different genes that could be knocked out would knock out a lot of metabolic pathways, probably not resulting in a living cell. So we decided the only way forward was to actually make chemically in the lab this chromosome so we could alter its gene composition. And that's how the whole notion of synthetic genomics was born with that relatively simple notion. So we started out with two primary questions. Can the chemistry actually permit making these incredibly large biological molecules accurately in the lab? And if we make it, what can you do with it? DNA is an inert chemical. Can we actually boot it up into a living cell? This is comparing the two areas. So the red line here is reading the genetic code, and the blue line is our ability to synthesize DNA. It's actually 5 orders of magnitude slower right now for writing the genetic code than it is reading it. But this is changing exponentially, basically just over the last few months. And I think writing the genetic code will soon catch up with our pace of reading it, even though it's still changing. Now, we thought this would be relatively simple. So there's machines called DNA synthesizers that can make very short stretches of DNA called oligonucleotides. We make pieces that are about 50 letters long. And we thought if we just make a number of pieces and they overlap each other, that they would just all go together and we could make a larger piece of DNA. It turns out you can, but the process with these machines for synthesizing DNA is very inaccurate. It's a degenerative process, so the longer you make the piece of DNA, the more errors there are with it. So we set out to do an experiment trying to make the Phi-X174 genome. Phi-X174, for those of you who don't know, is a bacteriophage. It's a virus that kills bacteria, particularly E. coli. And it was one of the first viruses that was extensively studied. In fact, it was the first actual genome of any type that was done by Fred Sanger. We chose this because, in fact, its genome is very intolerant of changes. If you change the genetic code, the virus cannot reproduce. So we developed some new techniques for actually accurately writing the genetic code where we can repair the errors in real time as we're making it. And for us, even though there had been an attempt by another group to make the poliovirus, which was slightly larger than this, it only had 1/10,000th of the activity because of all these errors in making the genetic code. We went from the genetic code in the computer, designing the pieces so that they would go together appropriately, use this new process for error correction. And we ended up with a piece of DNA exactly the right length. And when we sequenced it, it was exactly what we had designed and were trying to make. The exciting part was we took this piece of DNA and inserted it into the bacteria E. coli. And what had happened was E. coli recognized this as a piece of software and started making viral particles. And true to form in nature, when the viral particles were released from the cell, they turned around and killed the bacteria that had made it. So this is a process we see all the time in nature. I was just speaking to oil executives, and I said they clearly understood that process. But this was pretty exciting, of just taking a piece of DNA and having it activated, making viral particles. So we view this as the software actually building its own hardware. And this is an important concept as we're trying to go forward in this field, that even most people that are working in this area have not truly grasped the implications of this, that we don't have to design life from scratch. We just have to design the software appropriately. In fact, we've gone to scale this up. Our plan was to make pieces that were viral-sized pieces that we would then put together to make an intact chromosome. We looked for a number of ways to do this. We'd been working on it for over 4 years. And there's a process in nature called homologous recombination. This is the paper that you might have read about that we just published, where we in fact made this entire chromosome. It's the largest man-made molecule of a defined structure. To print it out one letter at a time at 10 font with no spaces, it takes 142 pages just to print out the letter code for this structure. It's 582,970 base pairs or letters of genetic code, and it's over 300 million molecular weight. So the process that we used was not as simple as just making pieces go together, but it's close to that. But it started with the right design, and design now is a key part of biology. In fact, before we started even the design phase, we had to go back and resequence the entire genome, because the standard in 1995 was roughly 1 error per 10,000 letters of genetic code. Nobody thought about, when reading the genetic code, even with that seemingly accurate error code, that anybody would be using those sequences to reproduce the organisms to actually write the genetic code. And if you're starting with the genetic code in the computer, you can only make things as accurate as the information you're starting with. So we went back and sequenced the genome all over again and found 30 errors, which was the standard for where it should have been. But we know with those 30 errors, if we made this chromosome based on the initial sequence, it would never been able to be booted up. So we start with this digital information, and we design the pieces, thinking that we're now breaking this 580,000 piece into 50 base pair segments. The 50 base pair segments have to be designed so they overlap with their neighbor. And we start off by making pieces on the order of 5 to 7 kb, 5 to 7,000 base pairs, that each had to be designed so when they lined up with their neighbor, they would overlap. We designed some other unique elements into it. You might be able to see this on the top line. It shows the places in the genome where we inserted watermarks. We wanted to absolutely be sure that we would not fool ourselves or others by having a contaminant of even one molecule of DNA from the native organism. And this can be a fun part of this process. So we have a four-letter genetic code. I'm sure most of you know that we have this triplet code, or 3 letters of the genetic code for our amino acids. And we have roughly 20 amino acids, and there's a single letter code representing each amino acid. So we can write things in the genetic code using this procedure. And we use this, in fact, to label the DNA, and the team came up with some of the authors of the genetic code in the institution. We've read that people were very disappointed we didn't put any poetry or other more profound statements like one small step or anything like that. But we'll think about that more carefully for the next time. So all this went into design, started making these pieces. We put 4 of these 5,000 to 7,000 base pair pieces together to make 24 kb pieces. And at each step, we would grow these pieces up in bacteria E. coli to make large amounts of the DNA that we then sequenced to make sure it was absolutely accurate. Because not only were we trying to make the end product, which we could have done much faster, we were trying to make absolutely robust methods so we would understand if errors crept in, where they came from. We combined the 24 kb pieces to make 72 kb pieces. And the prior world's record for the largest piece of DNA made was around 31,000. So all these pieces at 72,000 greatly exceeded what had been done before. Then we put those together. This looks like a basketball playoff, perhaps, to make what we called quarter molecules and half molecules. But what we found is we exceeded the limits of cloning in bacteria, and we switched to another system. We switched to yeast to put these together and to grow them up. In fact, we had always envisioned that we'd put these pieces together by this process called homologous recombination. Basically, cells in nature do this all the time with repairing their DNA. In fact, this organism, Deinococcus radiodurans, which is pretty ubiquitous on the planet and perhaps elsewhere, can take 3 million rads of radiation. Its chromosomes get blown apart with a couple hundred little pieces. Then, as long as it's in water over 12 to 24 hours, it remakes its chromosome exactly as it was before. So here's an actual picture of it after 1.75 million rads of radiation. I recommend you not try this at home, because we as a species can only take a tiny, tiny fraction of this amount of radiation without being killed. We can't do this with our human chromosomes. And so this is a pretty stunning process. It turns out this cell is not unique. We have thousands of species on this planet that can do this. These cells can be totally desiccated. They can be dried out. They can be in a vacuum. They can accumulate this ionizing radiation for a very long period of time. We don't actually know how long, but the speculation and calculations we've done is it would certainly fit into the Long Now Foundation, because it's tens of thousands to millions of years if the organisms even had slight shielding in a comet or other material. We know this organism can survive in outer space. It reaches an aqueous environment, and it reassembles its genetic code, and it can start replicating again. So we thought these mechanisms that we're trying to isolate from this organism would be great for assembling the little pieces of the genome to put it back together again. But in fact, we found it was even simpler than that. It turns out yeast, which is used for making bread and beer and wine and all these good things we like, can do this on its own with foreign pieces of DNA. So while we were trying to grow up the pieces in yeast, we found that if we designed the pieces correctly and put them in, it would assemble those automatically into the larger pieces, into the intact chromosome. So that's how we ended up with the entire 580,000 base pair piece that we sequenced, and it's down to 0 errors. This is actually a picture of it. You don't need an electron microscope because this molecule is so large. This is just looking at it over a 6-second period. So you can actually see it's a circular piece of DNA. And so it was pretty exciting for us to be able to actually visualize it. Well, how do you boot up a chromosome? You saw with the virus, all we had to do was insert it in a bacteria, and the bacteria could start reading that software and producing things. We think it's a little bit more complicated with whole bacterial chromosomes. Initially, we thought we would have to try and remove the chromosome from a bacterial cell and add in this new one to replace it. It turns out that's very hard to do. So what you've heard about with mammalian cloning works very easily in eukaryotes, where there's a defined nucleus that's easy just to cut out of a cell under a microscope, lift it out, and put in a nucleus from a different organism or a different cell from the same organism. With bacteria and archaea, there is no nucleus. The DNA is part of the cytoplasm of the cell, so we have to use a little bit more ingenuity to do this. And last year we published what we think is the key technique of transplantation. That's pretty stunning in its own right, because we actually, by putting in a new chromosome into a cell that had a different chromosome in it to start with, we completely converted one species into another. So it was an absolute 100% transformation. We used two closely related mycoplasma species, roughly the same distance apart as we are to mice. We isolated the chromosome from this mycoides cell, and we wanted to make sure we could get down to just naked DNA. Chromosomes have a lot of proteins associated with them, so we treated these pretty harshly with digestive enzymes to digest away all the proteins. And then we added back a few additional genes to this chromosome. One set of genes, the LAC-Z set, for example, will turn the cells blue, so it makes it easy to identify them. And we also added in a set of selectable markers, so we could select for cells just with this transplanted chromosome. And we put this chromosome into a species called a capricolum. Now, here's a wonderful graphic. I'm sure you'll appreciate the sophistication of this. We transplanted the chromosome into the cell. In fact, we thought we would end up with this situation, a cell with two chromosomes in it. We see this all the time in nature. So all these people that make arguments against evolution because we know that you can't just get a point mutation in one piece of DNA unless it's for the wavelength of light your photoreceptor sees, that's going to change into more complexity. What we see in the real world is we see chromosomes moving around where you can add 1,000 new traits to a cell in a second. Cholera, for example, most people thought there was no point in decoding the cholera genome because it was very closely related to E. coli. But when we read the cholera genome, it turns out it didn't have just a single chromosome. It had two chromosomes, and it looked like this. They were very different from each other. Obviously, it had taken up a chromosome from another species and added it to its repertoire. Well, we didn't want this situation. It turns out the capricolum genome is very unusual in that it doesn't contain any genes for restriction enzymes. Restriction endonucleases are the molecular scissors that cut up DNA, and it's how cells protect themselves from this foreign DNA coming in. In fact, the chromosome that we put in did have a restriction enzyme. As soon as it was in the cell, it got expressed, and it recognized the original chromosome as foreign and cut it up into small pieces, and it got digested. So we were left with cells just with the transplanted chromosome. And over a very short period of time, we ended up with these blue cells that all the characteristics of these cells were what was dictated by the transplanted chromosome. Every protein in the cell changed from that with the original species into that coded by the mycoides chromosome The membrane changed, everything changed. We could isolate the DNA, and it was only what we had transplanted in. So this is true identity theft at the ultimate level. And fortunately, most of us have mechanisms to protect us against this, and most cells do with these restriction enzymes. But they give us very powerful tools to try and do this in the future. So we know we can take a chromosome, we can transplant it, we can completely convert one species into another. So we're in the process of doing this right now with the synthetically made chromosome. We hope this will happen very soon. These experiments go very slowly because the cells grow slowly. So it takes about 6 weeks once you do a transplant to see whether you have viable cells. But we should be seeing that hopefully in the very near future. That will complete the trilogy that we've been putting together on this, although it's clear now just from the transplant experiment that we know this will work. And to me, it's more of a technicality if it works with synthetically made DNA versus DNA out of a cell, because it is the same, its sequence is the same, but I think it's still important for the proof. But it transforms us into a new era of now being able to alter cells by simply rewriting the genetic code. So the next steps and why. I'm going to walk you through quickly some of the reasons we're doing this and where we're trying to go. But a lot of people, when they think about this and write about it, they think we're redoing Genesis, and we're not. This is much more like a new version of the Cambrian explosion. We're dependent, as the title of this lecture said, on the 3.5 billion years of evolution. Also because we're here as humans because of that, and we're using organisms that have evolved over a substantial period of time. What's different is we don't have to redo all that, because if we just write new molecular software, we can now start at that point and go in an infinite number of directions. Why do this? One of the reasons is trying to deal with the future of our species on this planet. Over the next 40 years, we're going to go from 6.5 to 9 billion people. That's a huge change in a short period of time. I try to put this data in context for myself and hope that it leads others to be able to understand it. So I was born in 1946. There's now 3 people on this planet for everybody that existed in 1946. So if you think of having 1/3 of the number of people in this room, in 40 years it'll be 4 people for everybody that was on the planet in 1946. We're having trouble providing food, clean water, energy, housing for the 6.5 billion. We're going to have even more trouble trying to provide it for 9 billion people. We're changing our environment quite dramatically by burning billions of years of biology in the form of oil and coal. This is now from 2003, so over 5 billion tons of coal, billions of barrels of oil that we just take out and burn, and the CO2 goes into the atmosphere. This slide is out of date. It needs to be changed again the 3rd time in a year. This number is now 4.2 billion tons of CO2 that we're adding and stays in the atmosphere each year. That number is accelerating. The ocean sink, which is the largest sink, as you can see, that can take hundreds of billions of tons of CO2. Many people think it's saturated. And deforestation is also contributing as we try and strip more land to make more food and fuel. Everybody has seen this graph and the steady increase in CO2. So what can be done? There's a dramatic revolution that's taking place in the industrial world, equivalent maybe to the first industrial revolution. Companies like DuPont, which were basically built on using oil as its raw material have now had to switch away from using oil. They're switching to sugar, which maybe won't be any more sustainable, and we'll talk about that in a second. But they spent 10 years and over $100 million altering E. coli to do a simple reaction, to take 6-carbon sugar and make a 3-carbon propane diol molecule. I don't know if you can see those, it looks like large beer kegs. Those are actually 4 6,000-liter fermenters that they grow up literally 100 tons of this bacteria at a time in a batch and make propane diol from sugar with this engineered bacteria. They can do this cheaper and faster than they can with chemical conversion with their chemist. This has certain downfalls. After each batch, they have to go bury those tons of E. coli in a landfill because they're not allowed to burn it. But even with all these limitations and the cost of sugar, they claim their new polymer that uses this chemical will be the first multi-billion-dollar biotech product that's not a pharmaceutical. So metabolic engineering is being used. My new company, Synthetic Genomics, has a deal with BP to try and use biology deep in the earth to stop mining coal by biologically converting that coal into methane. We were quite surprised a mile down in the earth when we took the first sample. There was more biological diversity there than we found in the ocean. Just a simple microscopic field was teeming with microorganisms. This is a piece of coal that we have in the laboratory. All those little tiny things are the bacteria that live off of coal as a substrate, and we have other bacteria that convert that substrate right into methane. It's a little bit dark, but maybe you can see the methane bubbles coming off the coal in the bottom. This doesn't stop taking carbon out of the ground, but it's about a 10-fold improvement over mining coal and burning it. And the coal reserves are extensive enough that this could maybe get us as a stopgap until there's a new economy. But we've been going in the wrong direction already, in part because of lobbies in the government. Corn to ethanol just is not going to get us there. It's a negative carbon balance. It's been heavily subsidized by all of us. And the numbers are pretty stunning. It looks like a huge amount of ethanol is being produced. And we actually look at the numbers on the order of 6 billion gallons. There's now, I think, 160 plants around the country. 6 billion gallons sounds like a lot until you look at what we use. So this is our transportation fuel, 140 billion gallons of gasoline, 45 billion gallons of diesel. So 5 to 6 billion gallons of ethanol basically does nothing for this equation, especially when it produces more CO2 than it captures. It's competing with farmland. Food prices are going up because of this. Corn prices have doubled in the last year. So this is just the wrong experiment, taking us very much in the wrong direction. Now, we're not short of energy on this planet. 120,000 terawatts of energy arrives here each day from the sun. If we could capture that at 1% efficiency, we'd only need about 500 million hectares. I've argued this is a great use of Nevada. And nobody's ever argued with it. They haven't come up with a better alternative use. We could just from sunlight produce energy. In fact, deserts and seawater don't compete with food. We're in fact working on, I'll show you in a minute, what we're calling a 4th generation fuel, starting with CO2 as the feedstock. So if we can start with CO2, either from sunlight or in the methanogenesis pathways. We skip all this of use of farmland for producing fuels. We have two fuels that are pretty close to going into test production. We call them second and third generation. They both have the downfall of using sugar as the starting material, but they're far superior to ethanol. They don't mix with water. They have very low freezing points. They can just be flash blended. They can go through pipelines. They have a much higher energy density to get people going in a different direction. But the one that we think we'll have in about 18 months, the fourth generation fuel, starts with CO2 as a feedstock. Now, if we can really use CO2 as a feedstock, it does several things. When you consider the efforts for CO2 sequestration, we can just take piped CO2 into reactors, biological reactors, and make fuels such as octane directly from carbon dioxide, either using sunlight as the energy source or molecular hydrogen. You won't see any energy plans incorporating these types of ideas. We go forward by extrapolating from the past, from linear ideas and linear thinking. There is no planning for disruptive technologies. But in fact, if we're going to change anything in anybody's lifetime here, we need disruptive technologies. At the Davos meeting this year, the conclusion was 40 years from now, biology and all alternate energy sources will maybe only make a few % impact on using oil and coal, because there's so much vested interest. There's a new fueled power plant coming online every day. These cost billions of dollars to build. So it's only if there's an economically viable alternative that doesn't compete with food that has a chance to change anything in the near future. And I don't think we want to keep adding indefinitely CO2 to the atmosphere. So we have 10 million genes in the databases right now. We're going to double that number again this year from the Sorcerer expedition to 20 million. These are our design components. The entire electronics industry only had a few handfuls of design components, and look at the diversity that we have there. We have tens of millions of starting components that we can build. And now we've built a robot that, instead of making one chromosome per 4 years, we think we can get up to making thousands to millions of them a day, and in random combinations. So we're calling this new field combinatorial genomics. So just think of it. If we can basically start with design, take these tens of millions of design components, just make the DNA, transform that automatically into yeast or some other vector, and screen for chemical production, for octane production, for antibody production. Whatever you screen for will be the selection that happens. It will change the pace of biology in unimaginable ways, just like I don't think anybody could imagine in the 1950s this computer, this laptop computer with close to a terabyte of storage on it, doing more than giant rooms could. This is a new area that's going to be driven by people's imagination. This is sort of a software that we're working on to make the new software, thinking of this as a true design phase, trying to come up with new fuels, new chemicals, and so it's an exciting time as we try to scale this up with remarkably small teams. This is a different aspect of the Industrial Revolution, in that it doesn't take armies of thousands. In fact, that's what we've done from the beginning with reading the genetic code. The yeast genome took 10 years and 1,000 scientists. We reduced that project first to 4 months and now to hours and we're doing the same thing now going in the other direction. It's clear we'll be able to have a huge dent in the first bullet here of increasing the understanding of life, because we need to use empirical methods with the huge data sets that we have. There's not enough scientists to ask reasonable questions to work out the biology of what's been discovered to date. But I'm more optimistic. I'm hoping within 15 to 20 years, we can really have gone a long way for replacing the petrochemical industry. Hopefully, biology will be a major source of energy. And bioremediation, if we can capture back CO2 and recycle that into fuel, we possibly even start to undo some of the damage that we've been doing for the last 100 years. And we're also looking at this in terms of every type of chemical and looking at making, for example, combinatorial antibodies so we can have instant vaccines for every variant of the flu virus, for example, instead of being a year late like we are again this year. This field is unusual, and this is my last slide, by the way. It started with us asking the ethical questions before we did the first experiments, not after we thought, gee whiz, we have a lot of potential here. The results of this team at the University of Pennsylvania published their review in 1999. They thought we were proceeding with the right questions and the right technology. There was a caveat about being concerned about biological warfare. The Sloan Foundation has just funded my institute and MIT for the last couple of years to do a complete review of the risk and the benefits. That got published at the end of last year. And there's ongoing discussions. A new body in the federal government got formed when we published the IX-174 genome. It got reviewed at the White House. They had to decide whether to try and make our data and processes secret or published in the open literature. I think it's one of the few good things that the Bush administration has done in science is they went and opted for open publication. But they formed a new branch executive committee that has people from every branch of the government reviewing this type of work. We've had public discussion ongoing basically since 1996 on this. We're trying to take it one step at a time, but it's about to expand in an exponential fashion. And I think it's going to be an exciting next 10,000 years. Thank you very much.
Or 10,000 hours, whichever comes first. Am I alive on the mic? Great. I had a question before I get into ones from, thank you everybody. How do you decide what to work on next? I mean, the instrumentation keeps coming along, new tools keep coming along. Do they push you into new opportunity areas? Or do you work back from kind of the most important threshold type needs that you see in human affairs and work back from them as to what you do, or something else?
Well, it's much more the latter. We try to ask the big questions, and we try to drive the development of the technology so we'll have the tools to try and ask some of those. I mean, it'd be wonderful now if we could sequence a human genome for $1,000 in a few minutes. But I think we're creating the need for that and driving that forward. So the same with these tools. It's taken a long time to develop the synthetic capabilities for DNA. The next stage is now that we have these robots are going to take us order of magnitude further. So it's the ideas and the directions are driving the technology formation.
Here's a question from Pam Strayer. Where are you? Wave your hand. Hey. If you were advising a 16 or 20-year-old on what to study to be part of the next generation of health and science, what advice would you give them, and what new kinds of careers do you see coming out?
Well, I think one of the exciting things about what we've done, and maybe why a small group has been successful, is there's we have truly multidisciplinary teams. Some of the biggest breakthroughs we have been in mathematics, new algorithm development, new computer computational approaches. Obviously, the tools that my friend and colleague Ham Smith and Clyde Hutchinson have developed in terms of cloning and manipulating DNA, just straight engineering. There is no field of science that is not applicable to this. Even astronomy, my most exciting new number that comes out of astronomy is in our own galaxy, we have 100,000 Earth and super-Earth planets, all likely to be in that sweet zone that could be compatible with life. That's just in our galaxy. So when we think about the ubiquitous nature of life, we're pretty silly to constantly think it centers around human existence.
What's your bet on whether life is going back and forth between Earth and Mars and other places?
Well, it's definitely being exchanged. Where it originated, we don't know. There's roughly 200 kilograms of material exchanged between Earth and Mars each year. If that's not carrying organisms like Dinococcus and other things that can easily survive, I will be the most surprised person on this planet. So I know we'll find indications of life on Mars. It could be just because we've contaminated it, but it's possible it's contaminating Earth with a different set. We only know about 1% of life on our own planet, so we have a ways to go to understand that.
Well, you're, as I understand it, starting to sample shotgun sequence whole populations in the air and all the way up to the stratosphere.
Yeah.
What do you find as you get farther and farther out from the surface?
Well, we're still working on the data on the higher levels, but perhaps the most exciting thing is we're starting to look at the gels with space dust, because we have techniques now where we can amplify from a single molecule of DNA. And there's nothing more indicative of life somewhere else than large macromolecules of DNA. So I don't know if from the gels we'll be able to find any, but we're looking for the first time. But I think we've talked about setting up sampling arrays on the space station. We've contaminated near-Earth space pretty extensively, not just with junk, but with microorganisms to an extreme. I think the most exciting story, my brother's here, works with NASA. He sent it to me. I'll probably get him in trouble for telling this story, but next time you see a shooting star, so in the space station, they used to just pump out the commodes into space. And they realized they were really contaminating space. So now they pack them into the waste material into stainless steel containers and launch it back into orbit. And so when you see a shooting star, that's good shit from space, man.
One of our previous speakers was Rusty Schweickart, one of the astronauts. He said that when he was in orbit, one of the most beautiful things you see in orbit is what they call a urine dump at sunset. Because it sprays out, and of course the sun blasts into it, and the Earth is dark below, and it's quite gorgeous. They don't do that anymore.
Well, unless they have disease, the urine should be sterile. But
The urine should be sterile. We were taught in the Army to use it on wounds, right? Because it's so sterile you can wash wounds. Didn't you do that in Vietnam?
No, I was in the Navy. We had better techniques.
More expensive. We always save money in the Army. Here's a question from Tyler Emerson. Were you against the publication of the 1918 influenza flu genome, or for it? Why or why not? You remember Bill Joy and one of our other speakers, Ray Kurzweil, were ferociously against that.
Yeah, it was absolutely the right thing to do, and here's why. Looking at that sequence, it was not obvious, even though comparing it to my institute is one of two major institutions sequencing most of the viral isolates from around the world. We could not tell, looking at the 1908 flu sequence, why it was so virulent. Mm-hmm. And we didn't also know how to make the right vaccines against it. So not only publishing it was the right thing, I think this new committee in the government approving it to be made through these synthetic genomic processes at the CDC. It's actually the first true Jurassic Park scenario, because it was RNA isolated from a soldier that had died in 1918 and also a woman that was buried in the permafrost. The RNA still exists in her lungs. The RNA was sequenced and the virus regenerated in the laboratory. We now know what parts of the virus lead to its extreme lethalness, and we can now screen for that, and also new vaccines are being designed against that. So it's probably going to be one of the most important breakthroughs in at least flu virus virology. I think Killjoy and others that really worry about these things, just take the wrong approach. And he's now funding all kinds of companies to do the same thing. So it's,
Larry Brilliant and Google.org and the guys who are paying close attention to where trying to head off the next pandemic. And the mantra there is early detection, early response. Is this stuff going to help early detection? Can we do monitoring much closer to the ground so that the detection of a pandemic doesn't begin with dead nurses in a hospital somewhere?
Right. Reading the genetic code certainly can. It's still amazing, though. I mentioned some of the issues with just doing scientific sampling around the world. There's a number of countries, particularly in Southeast Asia, and even China and Russia to some extent, that don't want the world to know how much flu they have. It's hard to get the samples out. So quite often you hear first about people dying than getting the samples. So the technology is there. If it was set up around the world, it would be easy to get instant detection. The problem is the political problem that I just mentioned and air transportation. We saw how fast SARS moved from Singapore to Toronto. Fortunately, SARS is not anywhere near as virulent as the flu virus. So we need to step up the surveillance. But I actually think the synthetic genomic techniques that we have are what's going to create vaccines in time. We're a year late and $1 short right now. So if this was a really virulent virus right now, we'd be in serious difficulty. The vaccine is only, 40% effective. There's just not time with the slow processes from even knowing what the current virus is to getting the vaccine in time. So we need to change that dramatically.
So instant virus means this week or this month or this hour? How fast can you do that?
I mean, it could be done in
instant vaccine, sorry.
Well, the vaccine part needs to be proven in terms of the detection. We can sequence hundreds to thousands of isolates in a day if we have the material. The rate-limiting step is getting the viral isolates from different countries right now. The vaccine side needs to be tested and funded, built at a scale to see if it can be done fast enough.
Question from Laura from the Long Now Foundation. You've been telling us about an incredible richness of really planetary scale biodiversity here. So should we still give a hoot about the spotted owl? Yeah.
Well, people care more apparently about species that they can see with their own visual acuity than this extreme diversity that we have in the invisible world. If you look at even modern textbooks in biology, they list diversity as mostly accounting things that we can see with our own eyes, not things that you need a microscope or a DNA sequencer to see and detect. We have all kinds of unique varieties of multicellular birds and mammals that I think are worth protecting. A lot of them going out of existence pretty quickly with as we change the environment pretty dramatically.
How would this technology help that process of preserving them?
Well, I'm not sure it's going to help the spotted owl, but it certainly, instead of having a canary in a coal mine, we have tens of thousands of organisms. For example, had we taken the samples out of San Francisco Bay before the oil spill, we could have told you exactly what changed as a result of that oil spill. and how long it really took that to recover in terms of the biology that's there. So that's what we've tried to do on a global basis, is get samples from the major coral reefs. Everybody loves the beautiful sandy beaches in the Indian Ocean and other parts of the world. These are a result of coral reefs dying. Just a few degree temperature change usually ends up killing coral reefs. Many people think it's because it's killing the symbiotic bacteria that can't deal with the temperature changes. But we now have the first measurements to at least try to do that, to track these. So it could be the ultimate environmental impact statement to see what is changing. It's probably good for offshore oil wells to really measure what's happening there. Perhaps these people that think they can get CO2 credits by seeding the oceans with iron. Most of us think those are some of the most dangerous experiments being done on the planet right now.
Say more about that. Why is that?
Well, the indications are it could cause irreversible damage to the ocean. And anything that has even a slight risk of that, why are we doing that? So a few groups can make money off of it. It's just an insane experiment.
One wants you to go along on that expedition to sample what happens before and after the iron seeding.
Well, what happens, so iron is a limiting nutrient, so you sprinkle a little iron on, you get huge algae blooms. Then the algae dies off and presumably sinks down in the ocean, and you get more, but it's going to deplete a lot of other nutrients. We have a problem off the west coast, particularly off of Oregon. There's these huge dead zones now, no oxygen whatsoever, so fish, mammals swim into there. They basically die from asphyxiation.
Well, that was caused by wind, not by iron.
By wind?
Yeah, it's changed the upwelling pattern.
Did Ronald Reagan say that?
No, the scientists in Oregon said that. Changing wind will be its own, maybe that'll happen with all this wind generation. Lots of people using antibiotics in their personal life, in the bathroom, in the kitchen. They head off all those germs. From what you're saying, there's no hope.
Well, not only no hope. I mean, the worst thing for us as a species is to try and live in a sterile environment.
Okay.
Bacteria are a key part of our physiology. So I think the notion that you can ever escape bacteria, of the tens, maybe hundreds of millions of species on this planet, most of them don't care about us. That there's only a few handfuls that truly infect humans. Even the ones that share our bodies, most of those are not pathogens. It's not understood at all the thresholds that change when all of a sudden bacteria we have associated with us start causing disease. But very few of them do. The trouble with these, the chemical killers kill the whole spectrum of life and leads to major mutations. That's why we now have, I think when you think about new emerging infections, the new completely antibiotic resistant Staph aureus, it's now killed more people in this last year than HIV in the US. It's due just to over-application of antibiotics. I'm not sure whether you contribute any of it to people trying to sterilize their bathrooms or their hands, but certainly to the widespread use of antibiotics in the medical community. It's not clear how rapidly that could spread. If you're not near death, you don't want to go to a hospital right now because you have a higher chance of getting a Staph aureus infection in the hospital than you do anyplace else. So it's.
Does your technology have a workaround for that?
Not right now. I mean, we can try combinatorial production of new antibiotics. We have this huge chemical diversity in the ocean that really has not been sampled. Most of the chemicals that bacteria produce are to kill other bacteria. Right. There's some organisms that we have that actually produce diesel fuel-like materials. They actually smell like diesel fuel, using that as an antibiotic around them. So we've probably sampled 1/1,000,000th of the antibiotics that are available in the environment that could be used. There's a complete disincentive in the pharmaceutical industry to make antibiotics and antivirals. Companies would far rather treat chronic diseases because they can make a much higher profit than treating acute diseases. So companies like Eli Lilly completely shut down their antibiotic department. We hired some good people out of that, but that's helping some areas. But we're going the opposite direction right now when we need new antivirals and new antibiotics more than ever.
So it sounds like one of the career directions, to speak to the earlier question. So, people are going through the jungles of the world looking for wonderful secondary metabolites that'll be useful against cancer and things like this. And it sounds like you're saying that the ocean's maybe even a better place to collect.
I don't know if it's better, but it's largely untapped. The chemical complexity far exceeds what our chemists can make right now. But again, the new biology, synthetic biology, gives a chance to replicate and change that, because we can just take the gene pathways, put them in cells, and make the chemicals in the lab. And so a number of people now are thinking about taking these pathways out of all the organisms that we've discovered and just making whole new sets of molecules that just greatly exceed the complexity of what we're currently using, because you can't make them chemically in the lab. So I think there's a lot to choose from. It's a question, who's going to do it? Who's going to pay for it? Is there the incentives and the ability to even get stuff out of the market? It's a huge hurdle. And I think maybe that's why the pharmaceutical industry is going for much higher profit molecules than antibiotics and antivirals.
When you speak to people in that industry, or the other day when you were speaking to people in the oil industry, what do you talk to them about? Do you say this kind of stuff? Or what did you say to the oil guys?
I told them that that disruptive technologies were coming, and that they not only need to prepare for it, but contribute to it to help it come faster. So they're part of the solution, not just part of the problem.
They're clapping. Did those guys clap?
It was a good response. I mean, I got out of there alive, so it's
a good sign. You were inveighing a moment ago against ballast swapping, that vast quantities of ocean bacteria are picked up in one seething part of the world and released into another seething part of the world and swapped back and forth. Do you see this as inherently harmful, or what?
It's hard to know, because it's been going on for so many years. It's certainly harmful when you get things like zebra mussel larvae, and that's had a huge impact in areas. Right. It's not clear whether all these new species on the East Coast are from that, or just people emptying out their aquariums. There's a certain selectivity with these organisms based on things such as sunlight, as I gave you an example of. It's not clear what happens and how much it changes the environment. Nobody's measured it before, so whether it's contributing to more diversity or less diversity, we know over and over again, when you get an imbalance, new viruses appearing in an environment that weren't there before, new organisms that become dominant, it has a pretty catastrophic response. Look at all the oyster industries around, the Chesapeake Bay used to be a huge oyster industry. San Francisco Bay used to be. There's now viruses that have wiped out all the oysters. Maybe it came from, nobody knows where they came from or even what they are right now. It seems like a silly experiment to do. And now most ships change their ballast water sort of progressively as they cross oceans. They also dump all their oil and things out too. But
it sounds to me like my fellow environmentalists should be jamming ahead in some of the technology you're talking about to or monitoring for all these various things, doing diagnostics like you were able to tell just by looking at the population of genes in a particular sample what part of the world it came from, what kind of water it came from. So presumably the same kind of things will go on with human cavities and all the rest of it. Not only be able to tell who, but what their problems are and all the rest of it.
Well, you can also tell where people have come from. It's been shown, for example, as we send soldiers to the desert, in a very short time in the desert, all the bugs in their oral cavity swap out to what's in that local environment. Nobody really knows what impact that has on long-term health or disease, and as this happens and the environments get changed again. So there are certain branches of our government that are using this information to try and track people. I was asked if we could use these techniques to track bin Laden by finding his DNA in sewer water. I said I was not the right guy to do that experiment. So
Trust me, there are others who would be glad to do it. And it's done a lot with animals, by the way. With a scat, you can tell a whole
That's
lot
right.
about where they were, what they do, what they eat, all the rest of it. And presumably that will increase with this. But also my fellow environmentalists are highly dubious about genetic modification in foods and cotton and crops and things like that. What's your view on that?
Well, I think it's hard to be right all the time in each area. It's we have a dramatically changing environment that it's really hard to realize. We all like to think life is nice and simple as each of us have known it. And that's why I try to use the number about the population and trying to personalize it. It's a very different world changing within a single lifetime. And you mentioned that in your introduction. We have to find a way to cope with the soon-to-be 9 billion people. The environment is changing. If we don't do it intelligently, people are just going to do slash and burn to grow whatever is the most profitable crop, either to provide fuel or food for their populations. That's what we see happening right now. So I think environmentalists should be the first to embrace these new technologies, not be lining up to try and stop them. It's going to take, I think, a little bit more thinking on the part of numerous groups to make that transition.
Well, there's a question, and people say, okay, a fish gene in a tomato is just too weird. That's not Darwinian.
But tomatoes are not Darwinian in the sense that people fight for eating their natural tomatoes. Natural tomatoes, a small green thing about the size of my thumb that's non-edible. It's really bitter. So we've been doing selection of species on this planet for a long time. And what most people think is natural has been engineered, usually blindly. Now that we know about the genetic code in the genome, if people are going to propose, let's just randomly mix these genomes together from these diverse species, let's see what we get. It's called agriculture. It's what everybody ate before coming here tonight. So I think we need to put it in the context of what the reality has been going on in the last century of randomly mixing plant, animal, bacterial organisms together. So what's so unnatural about a fish gene and a tomato? Both those genes exist in your genome anyway.
So one of the things I'm getting from you and from Hawkins and others working in this field is that genes themselves are pretty migratory. They wander from genome to genome and species to species and genus to genus and all over the place. Is this your sense of things?
Well, in fact, I was on a recent program with Richard Dawkins, and I told him that I've begun to think of the world totally in a gene-centric point of view, that genomes are just unique combinations of those. There's basically an infinite number of combinations. We know a few of them. We see the big ones, as I mentioned, with our own visual acuity. We have another several million, if not tens of millions, to discover on this planet. And if we use them as design components going forward, we can really harness that information to do something unique. The genome structure is important. Regulation is very important. when we go from single-cell organisms to having 100 trillion cells, there's a lot of key regulation that does go on. We're a long way from mimicking that, but we're using these tools to try and understand it. I think the sooner we understand it, the better chance we have of surviving here.
You want to move on up the complexity chain into eukaryotes and bigger animals in terms of
I think it's essential. As you know, when you talk about long now futures, many people have argued humans have two chances for survival. Let's find another planet to go contaminate and move there, or let's engineer humans to survive in higher CO2 concentrations or altered biology so we can survive on the mess we're making here. Those are the two long-term current views that many people hold for humanity. I'd like to think we have a third alternative of trying to change the environment before we get to that point. But certainly engineering, if you apply design theory to plants, and you just design a plant for a specific purpose that'll live in an arid environment, need very little water, maybe even live off of salt water, and produce useful feedstocks for fuels or food, that could be a pretty nice thing. Design in disease resistance, the kind of replication you want, et cetera. We are living machines. We are designable, and I think it will be, in the time course that your foundation looks at, it will be inevitable to engineer humans.
Great hush, Phil. Two more questions. One from J.D. Ross Leahy. You use a lot of computer metaphors in your explanations, booted up, software, hardware. What do you see as the main similarities and the main differences between computer programs and the organisms you work with?
Well, we don't have too many computers yet that can build their own hardware from their software. So I think dynamic, some people are hoping to get there. We're partway there in some assembly lines. Biological systems can do this very dynamically. You can actually build the computer from the software. I think it's an important difference, at least for now. I think the microorganisms, for example, can produce things on scale, copies of themselves on scale that can be used for industrial purposes. When you look at landfills, obviously we're producing computers on some sort of scale, but not necessarily to solve directly manufacturing problems. A computer can process information. The biological software, the biological computer can produce food, fuel, chemicals, who knows what. Plastics can be produced biologically off of plants. I think as we start down this road, even coming back to talk a decade from now and looking at all the things that people are trying to do through biological engineering will be pretty dramatic. If you look 1,000 years from now, we probably wouldn't recognize anything because it could all be biological machines, including computers.
Well, I guess there's another comparison that's often made between computer technology and biotechnology is the pace of self-acceleration that goes on. So Moore's Law, doubling its capability every year and a half or so. People like Rob Carlson say that the pace of advance in biotechnology right now is faster than that. One, is that your sense of things, and is it continuing to accelerate, and does that S-curve ever level off, or is it pretty indefinite?
Well, I had a fun discussion with Gordon Moore because I argued that DNA sequencing, reading the genetic code biology was changing faster than Moore's Law.
Right.
But he said, you're dependent on computers, so Moore's Law always trumps Ventnor's Law.
Until we get biocomputers, and then that trumps that, or quantum computers or something.
I think it's sort of like the stages of evolution of, we have this 3.5 billion years behind us. It's not going to take, even possibly decades to mimic what happened over billions of years and go dramatically further. So I think we're at a sharp turning point in biological systems as we can go just straight from digital code to biological code and harness that power to do new things. I think that's going to totally change the future shape of humanity, hopefully the future existence of humanity. But I think it's going to be a very sharp divide from the previous 3.5 billion years.
Well, that sort of raises the question of why you talk to audiences like this. You were just telling me backstage you give about 150 talks a year. That's quite a lot for a still very busy scientist. And so every time you're out doing this, you're not in the lab. You're not raising another round of money or applying another round of money. You're not doing the research you might want to be doing and catching up with other people's work. Why put this much time into basically public discourse?
Well, we've seen over and over again new ideas, new disruptive technologies really being either killed or set back dramatically. I think, Jim, your environmental friends talking about GMOs, that has certainly set back a lot of an agricultural revolution. The whole thing was handled badly all the way around in terms of government regulation. There wasn't enough of it. There wasn't enough scientific testing to really work out what happened. And a lot of these genes, as we talked about, do hop around in the environment and they can move. There wasn't enough thought given to that. But several years ago, I was meeting with the head of the German Green Party, and I said, when you put that you're either for or against biotechnology or for or against GMOs, I'll always come down on the for side. But if you try to have an intelligent discussion about how it should be done, it should have been done very differently with a lot more testing, a lot more thought, and a lot more regulation. I think as we go into a much more dramatic phase of actually designing biology originally from scratch, possibly, as it scales up, if the public is not thinking about it, if the government's not thinking about it in the lines of what it can do and what it can't do, this whole area could get killed off just as fast as GMOs have, or set back substantially. I would like, as I argued at Davos, that we need to try and change something in the next 40 years. And these arguments that it's going to be almost impossible to change anything, the only hope there is for disruptive technology. I see biology as one of the few areas that really has a chance to be a disruptive technology in the fuel production, CO2 capture. We don't want people to be scared away for the same reason that French farmers tried to shut down GMOs, either because of an economic argument. So we're seeing a little bit of an economic argument with the corn to ethanol being, appropriately attacked, but people want to throw out all biofuels now because corn to ethanol is a lousy experiment.
Right.
These are dangerous times that I think technologies have to be part of people's repertoire. It is no longer an option for society not to be dependent on high-end science. If we're going to survive as a species long-term, if your foundation's going to exist in 10,000 or a million years, or people will, we have to do something very different very soon.
Well, it's okay. You're talking to lots of audiences, large one here and an even larger one on the other side of the television camera. So it looks like you're looking for understanding from these audiences. You're looking for a kind of permission and even encouragement from these audiences. With computer technology, the technology went totally democratic. Anybody could do it. I assume once you have the $1,000 genome and other tools like that, which are fast, cheap, they will also be fast, cheap, and out of control. Yep. And so there'll be biotech hackers. And there will be people in involved in national security who will freak out that prospect, probably with reason. Killjoy will freak out that. Ray Kurzweil.
Well, he better stop funding all these new
won't know what to do
companies
about
then.
that. But it's coming.
Yeah.
So garage biotech, threat, menace, promise, delight, what is it?
Well, there are some people in this new broad field of synthetic biology, not synthetic genomics, but synthetic biology that are encouraging biohacking, and to some extent that is inevitable. The techniques and tools require a little bit more sophistication for building genomes and species, but it will become largely democratic. We want everybody to have their genome sequenced. Understanding that is the only way we're going to lower healthcare costs, getting into preventative medicine. You know that firsthand.
My wife's in the business.
That's right.
We're grinning.
She's been pushing this longer than most. The world's slowly catching up with her. So we want to get to that phase in medicine. We want people to think in terms of DNA, what it means and what it doesn't mean.
Okay, so you not only want their permission, you want their cheek swabs.
Well, I don't personally want them, but
but there's plenty who do.
You know,
and once that's out there, then that database of humanity is baseline data. It's all these things you've been talking about, understanding who we are, what's going on,
right.
what we're supposed to do with that knowledge.
And I think that there'll be two surprises on the human side. I think we'll be surprised, and I've written that with John Brockman's Edge, that we'll be surprised how much of a genetic species we are. And we'll
what does that mean?
That most, if not all, human characteristics and traits have a genetic component. And you know, most parents realize this. They see their kids and realize that they have unique personalities from day one.
Right.
They don't want to acknowledge necessarily that's due to genetics, depending on the parents and depending on the personality. But we are very genetic organisms. But also at the same time, we are a very plastic species, meaning we're very moldable depending on our environment. So to some extent, knowing what you're at risk for will help with preventative medicine and disease. In terms of the sociology, I think it probably won't matter that much because the environment is so important for us. But we will find out, and we'll find out on a broad scale. What I'm worried about with a lot of the startup companies that are measuring incomplete information is that we'll just get wrong answers for quite a while before we have the complete information. But eventually, science trumps ignorance. Depends on how hard the equation is. You know, the internet could help or hurt with that issue.
Well, we should call it an evening. Keep the science coming, and we'll keep interneting at your science and help every way we can. Thank you.
Great. Thank you.